Hi, and welcome everyone to our Precisionary Scientific webinar series, and thanks for joining us today. So I'd like to start by apologizing for a last-minute change. Unfortunately, Dr. Andrew Satterlee is unable to join us today, but we have the pleasure to have Noah Bell, who is an amazing research scientist in Dr. Satterlee's group at the University of North Carolina at Chapel Hill. He's going to be stepping in, and he's here right now. Now, Noah is mentored by Dr. Satterlee ever since he was an undergrad, and now he is a research scientist in his lab. And he'll be delivering the pretty much exact same talk that was planned. So our summary and learning objectives remain unchanged, and we hope that you will really enjoy this fascinating story of scientific research in actually real patient tumor tissues. So to tell you guys a little bit about Noah, he is an expert in the Screening Live Cancer Explants program in the Eshelman Innovation, and today he'll be discussing the talk, a living ex vivo platform for functional drug screening of patient tumor tissues. And we're excited to have him share this platform in advancing preclinical drug testing and shaping personalized cancer treatments with key insights into the use of our Precisionary Compresstome vibratome. So please join me this morning for welcoming Noah Bell. Noah, over to you.
All right, thanks, Abby. Thank you for the introduction. Like Abby said, my name is Noah Bell, and it is my honor and pleasure to be presenting this talk on behalf of Dr. Andrew Satterlee, who directs our program here at UNC. And I'm excited to tell you guys about our platform as a whole, and also how, as we continue to do more research to push our platform and our science forward, the compressed-tome has become an essential part of expanding. So I'll start by sharing a little bit about Andrew's own story and our motivation for doing this research. So this is a picture of Dr. Andrew Satterlee about 15 years ago. He was diagnosed with a rare heterogeneous primary brain tumor while he was in undergrad. And back then, one of the state-of-the-art diagnostic tools was the histopathology analysis. So the path report recommended chemo and radiation, but it actually didn't recommend anything more specific than that. He had a lot of different experts weighing in on the best treatment plan, but because the pathology report was so vague, they weren't agreeing on what the optimal treatment plan was. And a big part of Andrew Satterlee's story is this tension and this anxiety surrounding the lack of a clear treatment plan and not knowing whether the treatment plan they pursued was actually going to be effective or not. Fast forward a few more years. So these are two pediatric patients with Dr. David Cram, who is one of our clinical associates. They have the same brain tumor type, the same diagnosis, and when they have got a genetic screen, their tumors both had the same key molecular mutation, and so both girls received the same treatment. Now, thankfully, one of the girls had a tumor that completely disappeared when given this treatment, but the other tumor hasn't shrunk at all. What this indicates to us is that we need a better way to determine optimal treatments for each person. And there are different generations of tumor diagnostics, some of which we've already mentioned in these two stories. So the first generation was histopathology. That's what Dr. Satterlee himself received. And this approach looks at the physical characteristics of the tumor when put in a slide. And that can tell you things like the tumor grade, how much the tumor is invading, and things like that. Now, the second generation that probably a lot of you have heard about is molecular pathology. So looking at genetic mutations and trying to find key mutations that might indicate whether a specific tumor will respond to a targeted treatment or not. And this has been successful in some cases, but the mutations that can be used to guide treatment are limited in number, and they're also limited in efficacy. It's not like the presence of a given mutation gives you 75% or 100% confidence in every case that the treatment will work, as we saw in the case of the two pediatric patients. This emerging generation of diagnostic tool is called functional pathology. And it's called that because rather than looking at the physical characteristics on a slide or looking at the genetic mutations present in a tumor. Functional pathology looks at the ex vivo drug sensitivity of patient tumor cells. So whether this is accomplished by taking a sample of patient tumor and growing a spheroid, growing an organoid. There are different approaches, but they all focus on taking the patient's actual tumor tissue and testing it against different treatments and trying to understand how the tumor will actually respond, how the specific patient's tumor will respond to different treatments. And hopefully that will eliminate some of the uncertainties inherent in our understanding of genetics or our understanding of histopathology. And that generation of there we go. So that third generation functional pathology is the work that the team under Dr. Satterlee, the SLICE program, that's the work we're doing, is functional pathology. So we are using this method called a living tissue substrate. So we use a living tissue substrate, which allows us to rapidly engraft, treat, and analyze these live uncultured patient tumor tissue. And you can see a diagram of this here. So we, for example, for brain tumors, we would take a slice of living brain tumor, keep it alive in culture. Then we would take uncultured patient tumor that's been resected from the patient in surgery, and we put it on this brain slice, and we have found that by co-culturing the patient tumor with healthy brain tumor, we are able to sustain the tumor, we're able to maintain its characteristics and behavior, unlike you see in an in vitro platform where you're just putting patient tumor on a slide, on a glass plate, or unlike we see in other functional diagnostic platforms. And now, our first target, motivated by Dr. Satterlee's own experience, was treating brain tumors. And when we were making slices of brain to use in this platform, we were able to use a more standard vibrotome, and that was sufficient for slicing the brain. But as our research progressed, we thought, well, what if we could do this with other tissues? We thought we could make slices of lung. We could make slices of liver. We could make slices of kidney. And recently, we've even started making slices of spinal cord tissue to look at other central nervous system tumors. Now, we run into some problems when we try to use a standard vibrotome on these. So the liver has tougher tissue and it has some really tough vessels. So when you try to cut through the tissue, the vessels kind of get pushed more than cut sometimes with the standard vibrotome. The kidney has a heterogeneous composition, so it has a tough exterior, a softer interior. That poses some problems for making slices with the standard vibrotome. And the spinal cord is tiny. We're using a lot of mouse or rat models, so that tissue is really small. We have small samples, and it would be hard to make a section of spinal cord tissue on a standard vibrotome. And this is where the Compresstome has proved essential to the advancement of our research. With the Compresstome, we have been able to successfully create lung, liver, kidney, and spinal living tissue substrates that can survive in culture. And as we'll get to toward the end of this presentation, these slices are of such high quality that we are able to maintain them in culture and also put cell lines derived from different types of tumors on each of these sections. So we will return to the use of the Compresstome in our research, but first I thought it was important to elaborate on how we discovered the value of the living tissue substrate. So this was not just something that someone kind of came up with on a whim. We were trying to understand how treatments would affect tumor. And we found that in vitro models, just putting these tumors in a cell culture plate, that did not recapitulate the in vivo tumor characteristics. So on the left, you see here 3 different sections from an in vivo study where we planted 3 different types of cell lines in a mouse brain. And you see the top one's U87, and you can see for that U87 sample, there's a very distinct line between where the tumor is and where the healthy brain tissue is. So U87 balls up. But then if you go down to this bottom row, you see GBM8. GBM8 is very infiltrative. There's no clear delineation between where the tumor starts, where the brain starts. The individual GBM8 cells are infiltrating into the mouse brain in vivo. Now, when we did in vitro studies with these same cell lines, we actually saw the opposite behavior. So you see this left column with GBM8, over 40 hours, there's almost no migration. And that's reflected on the right. You can see that the average cell velocity for the GBM8 sample is very low. But when we look at the U87 cells, the cells that were not very migratory in vivo, in vitro, we see the highest cell velocity. You see that those U87 cells are really crossing over into that no man's land that was there on day 0. So we found that in vitro models do not really reflect the behavior of the tumor in vivo very well at all. But we tried to put some tumors on organotypic brain slices, or OBSCs. And we found that OBSCs can host tumors that better represent the in vivo disease. So remember how U87, the tumor shown here in yellow, had a very distinct boundary and really consolidated in vivo. And we see the exact same behavior on the brain slice. There's a very clear boundary between the tumor and the surrounding healthy brain tissue, and the tumor has clumped up into a ball. But GBM8 actually displays the same migratory behavior. And if we zoom in on this green GBM8 sample, you can see how invasive the GBM8 is. And we can zoom in even further on this extremely migratory part of the tumor. And you might notice that there seems to be a stripe of migration right through the middle of this tumor sample. And that actually follows the corpus callosum. So this is the exact same behavior we saw in other in vivo studies. The GBM8 would use this white matter tract to actually traverse across the brain into the other hemisphere of the mouse brain in vivo. And we're seeing that exact same behavior on the brain slice. So this was some of the initial data that suggested, well, maybe there's some merit to taking these brain tumor cells and putting them on organotypic brain slice culture. Maybe this is a functional substrate that recapitulates the in vivo tumor behavior. Now, this next data actually shows that tumors grown on organotypic brain slices better predict in vivo disease patterns. So that previous study was kind of a responsive, like, we don't like this in vitro data. Let's see if we can come up with something different. Now we're starting the study on the brain slice. We mix together different types of cell lines. So you see in this top row, we have U87, the very consolidated tumor, and GBM8, the very invasive tumor, mixed together. And then on the bottom row, we have LN229, which was kind of a moderately invasive, moderately condensed, kind of the middle of the road tumor, mixed with GBM8, which is the invasive tumor again. If we keep going, you'll see that by day 6, the U87 consolidates into this ball and really pushes all the GBM8 out to the edge of the tumor sample. And this, again, this is on an organotypic brain slice we're growing these tumors. But the LN229 and GBM8, while there is some difference in their morphology, they are able to coexist much better. And if you actually took a sample across, like a cross section of each of these tissues, you can quantify that. So you see that by day 6, the U87 and GBM8 have separated. U87 is in the middle, GBM8 is out in the sides infiltrating the rest of the slice. Whereas LN229 and GBM8, there is more LN229 in the core of the tumor, but there's a lot of coexistence there. So this is what we see on the brain slice. What are we going to see when we do an in vivo study? And you might not be surprised based on how I've kind of presented this, but the in vivo behavior is exactly the same. So when we co-culture U87 and GBM8 in vivo, after 10 days, we see a very distinct difference between where the U87 resides and where the GBM8 is infiltrating into the rest of the brain tissue in the mouse. And LN229 and GBM8, there is some difference in where they reside, but there's a lot more coexistence. And when we take cross sections of this data, we see the exact same behavior. Very high U87 signal in the middle, the core of the tumor. GBM8 is infiltrating. It's on the edge of this tumor spot, whereas LN229 and GBM8 are able to coexist much better. So this was really compelling data, really inspired us to keep looking into the merits of the organotypic brain slice culture as a platform for growing tumor cells. We also saw that tumors grown on brain slices predicted in vivo drug sensitivities. So there's another group in our lab led by Dr. Sean Hinchin. They look at a lot of stem cell therapies. They look at a lot of trail secreting stem cell therapies. And so we looked at the different sensitivities of tumors on the brain slice to this trail secreting neural stem cell. So on the left, you see here that U87 and GBM8 are both very sensitive to trail at a high ratio of trail secreting stem cells to the tumor cells. So when we put a lot of trail in with the tumor, we see sensitivity, but LN229 wasn't very sensitive at all. So what we did in vivo was we put the trail sensitive cell lines, U87 and GBM8, in with the trail resistant cell line, LN229. And the trail resistant cell line is in red. You can see on this top section of an in vivo mouse brain. And the GBM8 and U87, the trail sensitive lines, are in green. You can see that when there is low trail, both tumors are able to take up residence in the mouse brain. But when we have high trail, there's almost no trail sensitive cells left, but the trail resistant cells are still growing. And remember, the sensitivity to trail was actually determined by testing the tumors that were grown on the brain slice. So this was a further piece of data validating the idea that the brain slice could preserve tumor behavior and tumor sensitivity to treatment in a way that in vitro studies just couldn't. Later, we tested whether, instead of using these pre-existing, predefined cell lines that are used for reproducibility purposes across different labs, we wondered if we could take uncultured patient brain tumors and still grow them on the brain slice. And what we found is that brain slices can uniquely maintain uncultured patient brain tumor tissue. In this data, we see that the OBSC is able to host more patient tumor tissue. It's able to let this tumor tissue survive orders of magnitude better than just on a transwell insert or just in vitro. And we have some more data later showing just how effective the brain slice is at hosting uncultured patient tumor tissue that we got more recently. But you can see here, there are some cases where a sample of patient tumor that's been taken almost directly from the resected sample from surgery is able to survive sometimes 100 times better, sometimes 1,000 times better on the brain slices. This is the data I was referring to. So when we cryopreserve and thaw these uncultured samples of patient brain tumor, on this graph, there's a lot of data, a lot of different samples of patient tumor tissue. This is from a clinical feasibility study that we are still conducting. You can see that the brain slice in light blue is able to sustain patient tumor tissue survival after cryopreservation and thaw across, what is this? This is 11 different patient tumor samples, and we've gotten even more samples than this, but these are the only ones we used to compare against 4 different types of media. Brain slices always were able to sustain the patient tumor sample better than these other types of media in an in vitro context. So next, we wanted to see whether the brain slices could uniquely maintain the genetic profile of these patient tumor samples. So this diagram just shows our different control groups, our different treatment groups. So we snap froze some of the patient brain tumor biopsy. We cultured some of the patient brain tumor in vitro, so we made a cell line out of the patient brain tumor. And then we cultured some of the patient brain tumor on the brain slices. And all 3 of these groups, we did whole exome sequencing. And what we found is that the original tumors on the left, there are certain mutations shown up there in blue and in gray on the bottom. When we grow the tumor as a cell line, the patient tumor doesn't retain the same genetic profile. There are a lot of differences, and there are some differences in the key mutations that might have guided treatment when treatment is guided by these genetic mutations. But the tumor tissue, when engrafted onto the brain slice, was able to maintain its genetic profile, especially in those top 25 key mutations. So we found that the brain slice was able to preserve the genetics of the original tumor fairly well over the course of our protocol. And this slide really shows why we think the brain slices and healthy tumor, the living tissue substrates in general, this is why we think that using a living tissue substrate can allow for this maintaining of a tumor sample better than in vitro. On the top row, you just see some astrocytes, which are labeled with green fluorescent protein, GFAP. And there's no tumor present. So the astrocytes are not really active. They're not really doing anything. They're just all spread out. But in this bottom row, we see the astrocytes in green. Now we see the patient GBM in red. And in the bottom right corner, you can see how the astrocytes are really interacting with the brain tumors. The astrocytes, these neural immune cells, are interacting with the tumor. So not only does the tumor have access to a realistic microenvironment, the living brain slice actually provides the brain slice interacting with the tumor. So it's this bi-directional interaction. Tumor interacts with the brain slice interacts with tumor. And we think that this sort of environment is why the living tissue substrates provide such a suitable platform for the tumors to grow on, and such a suitable platform for maintaining tumor behavior and tumor morphology and tumor sensitivity to treatment. So we took all this data and we developed a standardized functional platform. We have published a paper about this, and we found that these brain slices were able to sustain the patient tumors and allow for testing of treatment efficacy. Right here, this is just a little summary of our protocol. So we are putting 2 samples of tumor on each hemisphere. So we have one hemisphere with one sample, one hemisphere with another sample. That's 2 tumors per organotypic brain slice. We're putting 2 organotypic brain slices in each well. And then we also have on the bottom right-hand corner this propydium iodide assay. So this is another benefit of using a living tissue substrate. Not only can we see what effect the treatment has on the tumor sample, we can also see what effect a treatment has on the organotypic brain slice itself using propydium iodide, which is a marker of cell death. So this lets us know what the target specific toxicity is of the different treatments we test. This is a diagram outlining our drug sensitivity score. So we compare the OBSC toxicity in red to the tumor toxicity to a given treatment. We look at a lot of different treatment response parameters, such as the EC50 or the area under the curve, the AUC. And we're actually comparing the therapeutic window. So we're looking at where is the treatment effective against the tumor, but not too toxic to the healthy brain tissue. And so this is a unique aspect of our platform where we're trying to find this therapeutic window where we have an amount of a treatment that will be effective against a tumor without causing too many negative side effects for the patient. And we're able to do this comparison across different cell lines and across different samples of patient brain tumor tissue. We're able to test a given patient brain tumor against many different treatments from our palette of treatments at our disposal, including some preclinical agents. And we are able to compare these drug sensitivity scores across treatments and across tissue types to try and find which treatments might be the most effective. And to give you an understanding of how this platform might be used in the future in a clinical setting, we actually have a case study where we have a patient tumor where the patient had the tumor resected, and then 6 months later, they had recurrence. So here you will see the genetic mutations, and the mutational profile is complex, but the genetic analysis does suggest that the tumor would be sensitive to temozolomide, or TMZ, which is a standard of care for glioblastomas, plus X-ray radiation. So this is what the genetic profile suggested, TMZ plus radiation. And what we see on our organotypic brain slice platform is that this particular patient didn't actually respond that well to temozolomide and radiation compared to some other cell lines, and even compared to some other patient tumors shown in red. So the initial tumor is in yellow here is PGBM, and then the recurrent tissue is PGBMR, shown in green. Yeah, and so that's what we see. This is just one case study, but it shows that temozolomide and radiation didn't work that great on the brain slice platform, but other treatments did. And we're even seeing this difference between like trametinib and azacitidine, which were not effective against the primary initial tumor, but were effective against the recurrent tumor. So the brain slice platform even offers this potential to get like this quick feedback and maybe even uncover certain changes in treatment that might be necessary when a tumor might recur. So we have talked a lot about the brain slice platform. Specifically, we did a lot of our validation on the brain slice platform, But now that we have these other tissue samples that we can get with the Compresstome, we've been able to start expanding our platform and our methodology to other tissues. So here you just see some different graphs of tumor lines grown on kidney tissue, tumor lines grown on liver tissue, and even a few grown on lung tissue, which is one of our most recent additions or explorations. And you see that these living tissue substrates are also fostering tumor growth over the course of a few days. So we think that this methodology of growing tumor lines, tumor cells on a living tissue substrate is not just useful for brain tissue. We think that it has potential utility for other types of tissue. And the Compresstome has really allowed us to explore those other types of tissue. So we can talk about a few of the reasons why the Compresstome has been so beneficial. I was just talking with a few of the researchers yesterday about like, why do we really like this thing? One thing is it's easy to change samples, especially with this new model of the Compresstome. You can see the little cylinder where you put the agarose gel that contains the tissue sample. It's become very easy to change samples, so you can keep making sections of tissue. There is now a magnetic blade holder, which makes it really easy to add a blade, to switch a blade when one gets dull. The new machine is faster and it's more lightweight, which is fantastic. And as you saw before, this Compresstome is generating healthy tissue samples. It's creating samples that are so healthy that they are able to sustain tumor growth on top of them and sustain those interactions between the tumor and the healthy tissue. So the Compresstome has really made the generation of these healthy living tissue substrates possible. Our lab, the Screening Live Cancer Explants Program, or SLICE for short, we just opened a core facility at UNC Chapel Hill. So we are generating organotypic brain slices. We also have some mesentery culture generation where we can test ovarian tissues. And there's a paper in pre-publication showing that. We can do all these sorts of different studies, and becoming a core facility has made collaboration just a lot easier. It kind of cuts through a lot of the like academic, bureaucratic red tape. So it just makes it very easy for people to get together and figure out how to develop a study and make it happen. So we're very happy about that. If you're interested in our work, we have a little website that shows some of our research. You can get to know some of our team members better. And we even have a video that was graciously produced by Eshelman Innovation that tells more of our story and talks more about our research. So you can scan that QR code on the left to get to our website. And then on the right, this is a link to the SLICE core page. And this core is hosted through Agilent. So if you already have an iLab account, or if you want to make an iLab account, the core page can show you more specifics about what different treatments or experiments that we do where we've kind of standardized the process. And that is the end of this presentation. I would like to thank you all for listening and thank the Compresstome Precisionary group for allowing us to present our work. And I would also like to thank Eshelman Innovation for funding our research and the Lineberger Comprehensive Cancer Center for collaborating with us. And I'd like to thank everyone in my lab, Doctors Sean Hinchin and Andrew Satterlee, for leading our group and for really fostering just such a healthy and encouraging work environment. It's really fun to work with these folks, and it's really fun to know that we're doing something meaningful and impactful. So anyway, that is all I have. So thank you guys.
Oh gosh, don't say that's all you have, Noah. This is amazing. And thank you so much for starting with that personal story of Dr. Satterlee and his brain tumor and his biopsy, and to be able to hear about this entire journey to where you are with now a devoted core facility is just absolutely phenomenal. So I want to open up the floor now. And before I do that, I want to say, please do send our best to Dr. Satterlee. And of course, very much thank you to you, Noah. We have a question. And Fabit, please feel free to unmute yourself and show your video if you feel comfortable. But if not, I'm happy to read the question. So the question here is, have you tested whether you lose cell types after snap freezing the tissue?
How do you test whether you lose cell types after snap freezing the tissue? That is a great question. And I I know that we have done some. I'm trying to remember the name for this process, but it's where it's a flow cytometry. There you go. I know that we've done some flow cytometry. We actually have a new grad student named Caroline, and she has done a fantastic job characterizing the initial cell populations present in the resected tumor samples and in characterizing what cell populations remain after freeze thaw. So that is how we have validated that the tissue population remains the same during the different steps of our protocol is flow cytometry.
Great. There are a couple of other questions, but I want to open up the floor to see if anybody wants to unmute or show yourself to ask those questions. If not, I will ask two more.
And Abby, would you like me to keep the screen share in case we need to go back to a slide or?
Absolutely, yeah. So one of them was going back to making brain slices, which are key for in vitro and then potentially in vivo studies. What are your best tips for maintaining brain slice viability? And then also a connected part, how long do they really typically remain viable to allow you for studies?
Yes, thank you for asking that. And that brings up a great point I forgot to mention earlier. So to answer the first part of the question, what are some best tips? Let's see. So when I started working as an undergrad, it was during COVID. I was like Zoom classes. I don't love this. And so I was one of the people working on making these brain slices. And I think one of the most important things was just being gentle. It really does make a difference, like how gently you handle the brain slices as they're being made on the vibratome. So making sure that you're transferring them to the transwell inserts with great care. We have seen that makes a difference, and we actually have some quality assurance data that our current brain slice technician has gathered over the course of months, and we see that as we develop this care in handling the brain slices, making sure we're not tearing them, et cetera, but also just being really gentle with them, it makes a huge difference in viability. And we have measured that with our propydium iodide assay. Another big thing is minimizing the amount of time between sacrificing the animal and getting the brain tissue into cold media. So when we're using the vibratome, we have ice around the little compartment that holds the brain slice media. And so that is one of the key variables that we've tried to minimize is if you were to start a stopwatch when you sacrifice the animal and then stop the stopwatch when you actually have submerged the brains in the media and you begin making slices, we think that that is a really crucial component of maximizing brain slice viability. And there's a little bit of cell death. You're cutting straight through the tissue, but we actually think that's a good thing because it imitates the resection cavity. You're just, in vivo, when you're taking these tumors out, you're slicing the tissue there too. So we have found that we're still able to see overall slice health, and we're still able to see, tumors growing healthily on the brain slices. And to answer the last part of the question, we have done studies where these slices last for 3 weeks or more. Our standardized protocol for evaluating tumor-specific toxicity only takes 5 days from resection to getting the final result. So we get the resection, the next day we generate the brain slice, we put the patient tumor on the brain slice, and then it takes 4 more days before we have our final toxicity results. So the vast majority of our work occurs on that time scale of 5 days, which is very important to us because in a clinical setting, it is important to be getting data back fast to guide treatment. But, and there are lots of people who, we didn't invent the concept of organotypic brain slice cultures. So there are lots of people who have maintained these for much longer, I think even on the order of months. But most of our work happens over the course of 5 days, but we've even done some studies where they kind of stabilize and stay pretty healthy in culture for several weeks.
Awesome. And then another related one, since we have a little bit of time, is there are also labs that for studying in vitro, in vivo kind of systems, they use brain organoids.
Mm.
So how, in your opinion and experience, how do organotypic brain slice cultures, which you guys use, compare to using brain organoids for studying tumor microenvironments? And the challenging question for you is some key advantages or limitations of each approach when it comes to functional drug screening.
Mm. Okay, that is a great question.
It's a big question for you, and you could probably give a 2 hour talk on this at least. So we're asking, just asking you to do a really hard thing to summarize it for us.
Well, let's see. I think that one thing that we like about the organotypic brain slice cultures is that we can make them quickly. I admit I don't know very much about brain organoids. I do know that they are another approach for studying, studying brain tissue and maybe even studying tumor dynamics. But I have heard that it can sometimes take a while to grow these organoids up. And so that is one thing that I think is a benefit to using the organotypic brain slices. And I think maybe one more pro for the brain slices is that because we are just taking these sections of brain tissue, we are preserving the structure of an actually developed or developing brain. And I'm unfamiliar. Perhaps there are some organoids that really precisely recapitulate the native brain structures. I'm sure that there are, but that is one thing we like about the organotypic brain slice is it's not even recapitulating the brain structure. It is just the brain structure.
It has the brain
Yes,
structure.
exactly.
Because it is the brain. That's right.
Yes. Now that kind of touches on one of the cons of using an organotypic brain slice is we are using a rat model. And we've gotten some questions because we're not using human brain tissue. And so perhaps if you could make an organoid using human neural tissue, then that could potentially be a benefit to using an organoid, is that they are is that you could use human tissue. I'm not sure whether that's possible or not, but I know that that's a limitation of our platform. And I think perhaps another limitation of our platform is that we are seeding the tumors on top of the organotypic brain slice. And I assume that maybe in an organoid model, because there's some three-dimensional stuff going on, you might be able to get a more complex three-dimensional picture of the interactions between the tumor and the brain. So those are my initial thoughts on like the pros and cons of the organotypic brain slice culture versus an organoid approach.
Beautiful. Beautiful. So I want to open up the floor one more time to our attendees who are still here. If you have any additional questions, and if not, please feel free to reach out to me. I would love to put you in touch with Noah and Dr. Satterlee's group and core facility. And with that, Noah, one more huge thanks for you. And this is wonderful. We will put also this on our website as well, Noah, so you might have more questions coming your way.
Amazing. We're ready. Thank you guys
All
so much.
right. And thank you for all of you for attending as well this morning. Have a wonderful week.
All right, you too.